So as we continue to consider variations in Mendelian genetics, what we next need to consider is sex-linked inheritance. So these are going to be genes on the sex chromosomes. Um, but as we and this is a great example as we continue to apply our understanding of Mendel's genetics with our much more recent understanding of chromosomes and DNA. So we've been talking about it the whole time, but again, remember that Mendel didn't know what genes were. That wasn't even a term at the time. These were just hereditary factors. But now we know we have genes that are located on chromosomes that are very specific locations. You can, in fact, tag a specific gene on a location on a chromosome, as you see in this picture here. The yellow dots are tags for a specific gene on these condensed chromosomes. So we have now this idea of the chromosomal theory of inheritance, right? So we know a little bit more about how this works. So what we can do is then superimpose our chromosomes on the results of Mendelian inheritance, and we understand on a very detailed level the law of segregation and the law of independent assortment, right? That the two alleles for each gene are going to separate during gamete formation, and that when homologous chromosomes line up on the metaphase plate, they're going to line up randomly. And so that's going to lead to our law of independent assortment. Now, there was a scientist named Morgan earlier in the 20th century who was studying genetics and using fruit flies, Drosophila, as his model organism. And you're going to hear Drosophila again and again and again throughout your career because it's very common and excellent, very well studied model organism. So these are fruit flies. And what he was looking at was eye color. And what he noticed in his experiments was that the eye color between a white and a red cross showed that the white allele was recessive to the red allele, but that no matter how many times he crossed it, he never saw a white-eyed female. So always the white eyes appeared in the males in the population. So this led to the idea that it was that this gene was located on the same chromosome that determines gender, which fruit flies determine gender the same way that humans do with an X and Y chromosome. So if you have an X and Y chromosome, you're a male fruit fly. If you have an X and an X chromosome, you are a female fruit fly. So the nomenclature for fruit flies is a little different. You have a little plus sign to indicate the dominance, the wild type, and the lack thereof, the W, to indicate the recessive version of the gene. So what we can see here is that cross that he made, but with the chromosome superimposed on it, again showing you that if indeed the recessive version of the gene, which is the W without the plus, is found on the X chromosome, all you need then is one version of the recessive allele in the males to produce a white-eyed male, right? So what we're going to see is that you have to do very specific crosses to achieve a white-eyed female. Now, like I said, humans have the same gender determination system as fruit flies. Again, an X chromosome and a Y chromosome. These two chromosomes are going to line up with each other on the metaphase plate, but there's going to be no crossing over because they're not homologs, right? So there is this sex-determining region on the Y chromosome that codes for the development of testes because the default for development in humans is female. So if you don't have a Y chromosome, you automatically go to the female developmental pathway. You can see the Y chromosome is not very big. There's not a whole lot going on with the Y chromosome. Its main purpose is just to kick over the pathway to form the male gender. Again, I want to remind you that this isn't the only way to determine gender in the animal kingdom. We have other methods, right? So just because we talk about X and Y chromosomes doesn't mean it's the only way to do it. It's almost never the case that something's exclusive. Now, what this leads to is going to be a slight modification to our typical understanding of inheritance. Because again, the other way to say this is males basically lose the protection of being diploid on anything that's on the X chromosome. Okay, so think about that. So diploid says I have 2 copies of every gene because I have 2 homologous chromosomes with identical loci, right? So if I have a faulty version of a gene, I have another chromosome that may have a healthy version of the gene that would quote-unquote rescue me from the faulty version. But if it's on the X chromosome, males never have more than 1 X chromosome, because if you had more than 1 X chromosome, it means you have 2 X chromosomes, it means you're female. Okay? So males only need 1 copy of the faulty gene to incur a recessive mutation. So what we're going to do is use a little bit of nomenclature here to kind of indicate a sex-linked gene. So what we're going to do is use the chromosome as a capital letter, and then use our standard nomenclature, capitals, superscript for dominant, and superscript lowercase for recessive. So if you had some gene here that's located on the X chromosome, in this case, you could talk about a homozygous dominant female crossed with a recessive male, right? Not homozygous or heterozygous. There is no homozygous or heterozygous for male sex length because he only has 1 X chromosome, right? And so then beyond that, the crosses, the Punnett squares play out exactly the same, but you have to put the Y in there as a placeholder. All right. So take a look at that and think about it. Yet again, what it lends itself to is this idea that males lose that protection of diploid, okay, being diploid. So it's easier, it's more common to see a recessive allele manifest itself phenotypically in a male because they only have that 1 chromosome. That's why more males are colorblind. More males tend to be hemophiliacs. Duchenne muscular dystrophy. These are all chromosomes, or they're all genes that have a mutation that results in this phenotype that's on the X chromosome. Right? Now, so you may say, well, that's really interesting. Does that mean that females then have twice as much of all the proteins that are on the X chromosome as males do? Like, how does that work out? Well, it doesn't, right? So there's a good portion of the X chromosome that basically becomes inactivated during development, and that forms what's called a Barr body. So in a female cell, really only 1 of those X chromosomes is truly available for gene transcription. Now, clearly not all of the genes on the inactivated or Barr body chromosome are completely inactivated. Otherwise, the sex-linked inheritance wouldn't work out like it does. But basically, it becomes a very interesting factor here because for Barr body formation occurs randomly during embryonic development. So the paternal chromosome, X chromosome, or the maternal X chromosome that every female has, either one can be inactivated. And if she's heterozygous for the genes on the X chromosome, then she's going to become basically a genetic mosaic with regard to that. This can be seen then phenotypically in some animals, for example, in cats. So the black and the black and orange fur color, this particular cat, is a gene on the X chromosome. So if patches of cells go inactive, so you get a cell that makes a Barr body that goes inactive that has the black color gene on it, then that cell, as it divides and grows into a portion of what's called the clonal progeny of that developing embryonic cell, will all be orange in color because that's the active chromosome, and vice versa. If the X chromosome with the orange gene goes inactive, so that inactive X chromosome is called a Barr body, and it leads to, again, some results that you have to dive a little bit deeper into Mendelian genetics to understand, but again, the basic concept and the basic rules still apply, right? So we'll continue with this idea of kind of superimposing our understanding of chromosomes onto Mendelian genetics as we continue to talk about this topic.